All right. Well, thank you guys for getting on. I appreciate it. I'm excited to have Dr. Eleanor Host from Perrysburg. And Doctor, if you could give a little bit about your background before we get going, but she's going to walk us through really science-based presentation on the immune system. And then when we get done, there will be a Q&A, so you can have questions. As a reminder, if you're on live or if you're watching this replay, this is a series. We're going to have a series of immune boosting tips and coming over the next really month. So we're going to have a good handful plus of videos that you're going to want to check out, you, your spouse, and please share them so we can be really informed about our immune system. Guys, our immune system response is everything to warding off all types of infections and cancers and whatnot. So with that, Dr. Eleanor, I will tune it over to you. And thank you for taking your time to do this. We appreciate it.
Yeah, absolutely. I love educating people. Am I coming through?
You are.
Okay, perfect. I want to make sure I didn't mute myself again. That would be a very short presentation. Okay, so we're going to talk about the immune system and a little bit about all the different types of testing that you have for COVID-19 to kind of give you guys some more background on that. And I am in Perrysburg. I'm a board certified family doctor. Gosh, look at that, since 1995. Wow. And also I do acupuncture. I'm just going to touch on that real briefly today. So if you guys want to hear more about that, just let John know and I can come back and talk with you all. All right, the immune system. So this is it in a nutshell on this one slide. So the biggest things that to watch for are the thymus, which actually does shrink up as you get older and produces fewer and fewer T cells, thymus T cells, and also the bone marrow, which produces the B cells, and we'll go over what those are. You also have your lymphatics, that's all the green part here, and then the little lymph nodes that we all know about when we get sick. And then your spleen is also an important organ in the immune system. It helps store cells. It gets rid of cells that are kind of near the end of their lifespan in the blood. And it helps get rid of some complexes. As your body kind of sticks the immune globulins to the invader, it helps clear those. A little bit more important in bacterial infections than in viral infections, though. So COVID's a virus, so we won't be talking about the spleen a whole lot. Okay, so there's the different types of cells. You have your T cell and the B cell. This is probably the only time in science where they actually have things labeled simply. Though the B's come from the bone marrow, and they transform into plasma cells, which actually are the cells in your body that make the antibodies that you have heard so much about lately. The T cells have several different kinds. One of the more important ones are the CD8 cells, and those cells are kind of like the ninjas of your immune system. They run around and attack right away, kind of at random. They don't really have a target. They just say, hey, you're an invader, I'm going to kill you. So they don't have a lot of checks and balances on them. They're not very specific. The CD4 cells actually are a little bit more specific, and they actually do influence the CD8 cells. So they kind of tell them, hey, dude, that's not what you're supposed to be killing. Go over there and kill that. There's a lot of feedback loops going on the immune system, and it's way more complicated than we're even going to get into today. Another thing to think about is you've got your innate response in the immune system, and these are what happens right when you first get sick. This is all the stuff that makes you feel terrible. When you feel achy and sore and tired, that's because your body has all these cells that are trying to get activated to fight off whatever just popped into your system, okay? After you've been sick for a while, the body's like, wait a minute, this is taking too long to clear. Then it starts activating the adaptive immunity, which is your B cells that make all the immune cells. And this is how vaccines work. So this is making immune globulins that are specific for whatever that invader is to help clear them quicker from your body. The thing with viruses that's really interesting is a lot of it does very much depend on dosage of how much virus you get as to how sick you might get, or even if you're going to get sick at all. Viruses themselves really are skating. Are they really alive, or are they really not really a live thing? Because they don't really live outside of a cell. They can't eat food like bacteria can. Live on a petri dish. You give a little bit of food, they grow into a colony, they like each other, they're happy. So that's how bacteria work. But viruses can't even reproduce on their own. They need another cell. They have to actually invade a cell to reproduce and make more virus particles. So when it's sitting there like on packaging, like we all heard at the beginning of, oh my gosh, it sticks to everything and you're going to be sick from everywhere. It doesn't live very long out in the environment. It needs to have cells to reproduce and make more of it. So if you're getting a little tiny dose of particles, your body might clear it right away. There might not be enough of that virus to get into your system and actually make you sick, which is why the mask wearing is so important. So that's the cells. The other thing that gets produced when you do get sick are the cytokines. You've heard the cytokine storm thing. That's actually the chemical signaler that your body actually makes to try to get more cells to come and fight the invader. And there's some question as to why some people have a worse response than others. Might just be your genetic code makes you make more cytokines. That's why some people who are younger might get really, really sick, even though we picture older people getting sick and having more problems. Some of those young people who get sick and have problems may have a problem with the immune system where their cytokines get too high. Okay, which is another thing going back to the adults, why the older people tend to get sick is their thymus gland doesn't work as well. So they don't have as many T cells, which is that initial response. So the older people get sick because the T cells aren't there. And the younger people might get sicker because for whatever reason, their cytokine response gets huge. The other thing we're going to look at here are we call antigen anything that's not supposed to be in your body that's considered an invader. And those can be bacteria or viruses. And some of the bigger cells, like this is the innate response that just happens automatically. They can recognize, hey, you don't belong in this body. I'm going to eat you up. And that's literally what they do. They go around and they gobble up things that don't belong. And in the meantime, the B cells have to kind of start going, oh, wait a minute, I'm supposed to be doing something here. Let me make some antibodies to help fight this off. And they actually stick to the antigen. So the antigens are these little green thingies, and the yellow particles would be the antibodies. And they make these big complexes, which are very recognizable to the other cells that eat them up. So that's how one way your body helps fight off viruses. And there's a bunch of different types of immunoglobulins that are made by those B cells, those plasma cells. It doesn't really matter what they are or what they do, but the IgA ones tend to be in your saliva and they line your gut. The IgG are usually produced later in an infection, and they're much more specific to that specific invader. And the IgM is kind of like the initial blast. You can see the IgMs are huge because they're really trying, they don't know what they're supposed to do, but like, oh my God, let me sample all these weird particles and I will try to capture as many as I can. So the IgM tends to go up first when you get sick with any kind of infection. And then the IgE, which is even more specific, will tend to stick around. The IgG are the ones that are going to stay for you for the rest of your life with any luck. Sometimes it doesn't work that way, which is why we get colds every winter. And that's why everybody's a little worried about coronavirus. It's going to be like all the other cold viruses where we have to get a vaccine every time, like influenza. But hopefully not, because there's part of the virus is very stable, and I'll show you that in a second. This didn't come out, but this is all the different kinds of immunoglobulins that your body makes. Why are antibodies important? Well, this is part of the thing they just approved by the FDA, which was doing the antibody serum. And what you do is basically you get sick and you make all these antibodies. Your body purifies, they drop from your blood, they purify it, and they're going to give you all those extra antibodies to somebody who's earlier in their infection, or maybe their body can't keep up with the infection they have, so we're kind of giving them an extra boost of antibodies to help fight it off. So that's kind of how that works. And I will say it is sort of questionable. They don't really have a ton of good data. They're still collecting more and more as we go on, but the FDA did give it emergency authorization, which means, well, it might work. It doesn't seem to be hurting, so let's just try to use it more and see what happens. I mean, you can tell that's not really the greatest treatment. I mean, I wouldn't want to think, oh my gosh, I think I have coronavirus. Let me go to the hospital and get a blood infusion. Not exactly the world's greatest preventive, so we'll have to keep hoping we get more things. And I have this really nice slide. This kind of explains what viruses are doing in your body. This is the coronavirus represented here. So what it does is there's actually these little receptor sites on your cells, and the receptor sites are kind of like a gateway. It's sort of like the guard in the parking lot. You need your pass card before you can come in here. And it's used in your body on all your cells because that's how it keeps everything, like my cell is a safe place and I can only let certain things coming in and out. What happens with viruses is they have proteins on them that actually accidentally happen to fit in the little keyhole. So they have a little key that accidentally fits there, like, aha, look, we accidentally broke the code. We're going to get into the cell. And the cell doesn't know any better. It's like, well, you matched the keyhole. You must be okay. You know the password. So that lets the virus particle in. And this is where it gets really cool, I guess, or bad, depending on how you look at it. I think it's cool because I'm a doctor. What can I say? So the virus particle gets into the cell, and it starts to actually unravel, and it opens up its contents. Now, virus particles either have DNA in them or RNA in them, which is like the building blocks of all cells, right? But they don't know how to do it. They don't use it themselves. They actually sneak into your cell. They break in, they unleash all of their stuff, and then they go to this thing called the Golgi Body. And this is the place in your body that actually makes protein. So it literally sneaks into your cell, takes over your factory. So not only is that cell no longer making proteins that your body needs, but it's actually making copies of the virus. And that's what it does. So it sits there, makes all these copies inside the cell. whoops, and eventually makes a new virus, and the cell actually will literally fill up with viral particles and eventually burst open with more new virus particles. So once you get enough in there and you start having more and more viral particles, you can see it becomes really overwhelming pretty fast for a lot of people. If you get a huge dose of virus at the beginning, it's going to be really hard to keep up. And using the parking lot analogy, that would be if you let one car in and, oh, wait, you're not supposed to be in here, you could go run and catch it pretty easily and say, hey, no, you gotta leave the parking lot, you don't belong here. But if 50 cars come in all at one time, I mean, and they're reproducing in the parking lot, it's really hard to catch up. So that's kind of why viruses can become overwhelming really quickly. There we go here. Okay, so this is kind of showing what's happening inside the serum of your body during an infection. So when you first get infected, this is what will happen. This is about day 1 in the beginning. The middle part where the antigen and the RNA are down and the IgM starts to go up, that's about a week with this coronavirus. That's about 7 to 10 days. So when you're getting the testing done, they're going to check when you first get sick. Within 5 days, that's when everything's really high for the viral RNA and the viral antigen. So those are the tests that you really want to get within the first 5 days of being sick. You know, if you test super early, like right away, like, oh my gosh, I was around Aunt Mildred yesterday and she tested positive, you know, and I would run right now. I don't have any symptoms. Should I run right out now and get a test? You know, that's the big question. And you could, but if you're not having symptoms, I usually tell people wait at least 5 days from your exposure and see if you get symptoms, number 1. Number 2, if you do have it enough in your body that's high enough, 5 days will be about right, because that'll hit you where you're starting to see a high enough level that the test should show something, okay? Now later on, if you were exposed, you didn't get sick, and you wonder, did I even have it? I don't even know. That's when an antibody test would be helpful. And that would be helpful 7 to 10 days after your exposure or when you think you might have been sick. And there's a lot of questions as to how long do those antibodies last, whether they be high enough to detect. And some studies are showing if you had a pretty good infection, your antibodies seem to last 6 to 8 months. If you had a very mild or asymptomatic case, they may be gone in 2 to 3 months. But again, that's still up in the air. That's just preliminary data that might change in another year when we have more information. So a lot of it is continually changing. Okay, so this is a test you could do. So we've heard of the PCR test. This is a really wordy slide, so don't even look at it. Don't worry about it. I will explain to you what it means. The PCR testing you can do for any type of virus or bacteria if we know their DNA or RNA, okay? So you can program, the computer will have like a little glue, like if you look down here at the bottom of the slide, it kind of will stick to different pieces of the DNA that match. But because there's such a teeny sample, what they actually does, PCR just means it's making copies and copies and copies and copies, and then attaches to this, and then it runs it through a sampler that says, this is how many copies we found, and there's a cutoff point. So if it sees lots of copies, then they say, oh, your test is positive. It doesn't see hardly any copies, your test is negative. So this is a test that will pick it up as soon as your viral RNA load is high or the antigen load is high. It should show up positive on a PCR test. And that's the one that they're doing with the big nasal swab. Okay, that's really unpleasant. Okay, because you need enough to get to copy. So the problems with this test are it takes a long time to run because you have to get the sample, send it to the lab, and the lab has to run it, and that takes some time. And you have to have, you know, it's like saliva. But you have to have the serum that goes with it to keep the test going. You need a test tube, you need the test kit. So there's a lot of hangups with doing these fast. So that's kind of a problem we had at the beginning of the infection with the coronavirus when the pandemic started, is we just didn't have enough of these kits. And that really slowed us down. So they have since developed other things. They can actually do antigen testing. Let me skip to this other slide so you see on here. They can do antigen testing this way, and they can also do antibody testing this way, which is easier because you can see you make a little test kit, sort of like a pregnancy test, right? You can do those at home. What they do with those is how all of these test kits work, is you're going to have a sample that you're going to put in there. These little orange dots would be like the virus, that's coronavirus. The paper is actually impregnated with antibodies to the virus. This one, the control, there's actually usually a solution that you're going to put on with the sample. If anyone saw me I was doing antibody tests the other day, you have to put a couple drops of this buffering agent that also has a chemical that will bind to the control, so the control will be positive. So if you don't have that, then the test is invalid. So as this drips through, this is capillary flow, all the little bits of orange, which is the particle, will start sticking to some of these antibodies, and then as it flows through across the paper, they'll actually stick and make the color change happen. But that's pretty cool. I always thought that was the neatest idea ever. Like once they figured out antibodies, like, oh, we can make test kits that do this. So that's a really good kit. And they have antigen testing now for that, which again, that's early in the infection, the first 7 days. And they have antibody test kits that do this, that are the last, like maybe 7 to 10 days after you've been infected. All right. And the vaccines are coming. They have a couple good trials, so here's fingers crossed that it's going to work for a lot of people. But again, that's all very early yet, so they're testing on such small sample sizes. You know, we all know, like, once, you know, drugs can get approved now, even with the FDA, and how many times does that happen? And then they go out in the community, you know, 10,000 people uses them, and they have to recall the drug because, whoops, we didn't realize it caused liver failure or whatever. So, you know, small tests are important, and we're going to need more. So, you know, I'm hoping we'll get a vaccine, maybe by the end of the year. But most people in the public are probably not going to be able to get a vaccine, probably for at least another year. I'm hoping we'll have it ready for next year's flu season, so we could do them both at the same time. But we'll have to see. And the big thing they're looking at with the antigen is they're trying to get the spike protein. That seems to be the one that, if we get that, your immune system attacks that right away. It can't attach to the cell, like you saw on that first slide or one of the earlier slides. Okay, so we're working on that. Now I'm going to get into more detail with testing. So take a big breath and clear your brain, because it's a lot of information. Okay, so sensitivity and specificity. You may or may not have heard these, because I know a lot of people are like, all those antibody test kits that they approved originally, they took them off. They said, no, these are terrible tests. We take them back. There are quite a few that do that. What you have to do for the FDA to approve a test now, you have to have 95% sensitivity and 95% specificity before they'll even approve it. Well, what does that mean? That's a statistical way of saying when you test positive, you have a true positive. And when you test negative, it's a true negative. So when you test positive, it means you really do have the disease. And when your test shows negative, that really means you don't have the disease. And that has to be at 95%. So it's pretty high up there. Imagine trying to get 95% on a paper you're writing. That's pretty high. All right. There's another graphical representation of that. If you look at this slide, the pink half of the slide with the open circles are negative tests. The left-hand side with the green and the solid circles is a positive test. And what you want to do is have very few false positives and a lot of true positives. Okay, that sort of makes more sense. And I will tell you, this is the hardest part to understand with this whole epidemic. When they first started, finally got more test kits, I'm like, okay, you can only be really sick people can get the test done. We don't want to test everybody. We really want to know who has the disease and who doesn't have the disease. But statistically, the problem you have is something called pretest probability. So here's another graphical representation. So what we're going to try to do is explain, here's a population. The green ones are the people who have the disease, and the red ones are the people who don't have the disease. And the classifier is the test, and then you want to predict how many are going to be negative and how many are going to be positive. All right. Now, in an ideal world, actually, this is not ideal. This is what we have now. So right now, I know it seems like, oh my God, everybody has coronavirus, but it's a teeny weeny percentage of the population. I mean, it's probably less than 5% still. You know, even if, you know, extrapolating out to assume those people probably had it and we never tested them, it's still pretty low. So this is what's happening. If you have a population with very low disease, that's all the red people, and maybe one person has it in that circle, and then you go through and test everybody, this is what will happen statistically. This is what, even if your test is very accurate, okay, but because your population level has almost no disease, you're going to have a couple people who are falsely positive, okay? So to limit that, when they first started having test kits, that's why they were saying we really only want people who look like they have symptoms, or people who are, you know, probably around it a lot, and they may be asymptomatic, but like healthcare workers who've been working in the ER, we should test all of them because they're around it a lot. So their population prevalence is a little bit higher, so that gives you actually more true results. So that would be more likely to be a true negative or a true positive, okay? What we are looking for is something like this, where most of the population has it. We don't really want most of the population to have it, but this will make your test super accurate. If more people have it, then the test is almost exactly perfect, right? All the positives are going to be positive, all the negatives are going to be negative, okay? So that's where it gets, like, people say, oh my God, my son had all the symptoms and he had a negative test, you know, so testing is stupid and I don't want to do it. Well, some of it's just the way statistics work. You know, and you have to just kind of like, well, as a doctor, what I do is I look at the symptoms, look at the likelihood that you have, and say, yeah, you probably have it even though your test was negative. And I want you to stay quarantined, because that's more important to your brain than to rely on a test result. And we all know that. All right, so I whipped through all that, probably a little quick, but, you know, it's a lot of information. I hope it at least made some sense.